are proposed to be very recently separated species. We basically look at genes on chromosomes. These numbers represent just different genes, and we look to see what order they come in. Here's what we see in one species. This is the one I work on, Drosophila melanogaster. We look at this species here. We say, okay, these ones match up, these ones match up. And then come out over here again. Yeah, these ones match up and these ones match up. But these ones in the middle are flipped over relative to each other. So they're still there, but they're in an inverted order. Okay? So basically, it's like this, then this, then this, then that. So we know in genetics that this sort of thing can happen. You can have a breakage in chromosomes, things can flip over, move around. Genomes are actually relatively fluid. Over time, they break, they rejoin, and reorganize themselves into new combinations. The evidence for that kind of movement is conservation of genes in a block. And that argument, or that observation, is something called synteny. We would say that these genes are in a syntenous block. They come together in a shared order in these two species. Okay. That's at a small level. So not only can we look within a species, we can trace these events between species, and we can sort of see how far this goes. I'm not giving a technical talk here, but if you want the articles that show this data, this is where I'm going to draw my next figures from. So this isn't my own work. Like any organism in biology, we have this group of organisms that are proposed to be related to one another based on morphological criteria. Okay? Here's the one I work on, proposed to be more closely related to this organism than to say that this one. These two are proposed to be quite closely related, and so on. So what you're looking at is a branching tree of speciation, essentially, over time, a proposed one. Here's what we see when we look at their chromosomes. So this is sort of just a high-level view of their chromosome complement, their genomes. Here's the X chromosome, for example. Fruit flies have an XY sex chromosome system like humans do. Here's the second chromosome, third chromosome, fourth chromosome. What you see is that, in general, these different groups, these different colors, represent large-scale blocks of synteny, genes that are in the same order in these different species. So you can see there's evidence here in this species that there's been an inversion right in here, right? This one kind of like relative to that. We also see some evidence that chromosomes have either fused or broken apart. Or in some cases, I don't know if they have it on this diagram, it might be on the next one. Oh yeah, here we go. We can see that here, this what's present as a small free chromosome in many species seems to have been fused with this chromosome here. Okay? So we have ways of tracking sort of large-scale movement of genes around in related genomes based on their sequence. Okay. Here's what it looks like if you look at it on a per-gene level. So each of these lines, now again looking at all these different species, each of these small lines is just indicating, okay, here's a specific gene, where's that gene found in the next organism? So between these two species, at the top, they're proposed to be quite closely related. What you can see is, for the most part, you have many genes that are blocked together, and then we have some large-scale rearrangements. Here's a big inversion, for example, other inversions. Here's one really big inversion, but the rest of the chromosome pretty much has everything in the same order. Okay? Now, as you get less closely related, based on, or at least predicted to be less related, based on those phylogenies that were generated on other criteria, we see that the measure of relatedness of where these genes are relative to one another becomes progressively more mixed over time. So this is an independent line of evidence, not just looking at the similarity of genes, but it's asking the question where we find those genes. And it seems that as we look at species proposed to be more distantly related to one another, we still find the genes in roughly the same spot. Okay, all the red genes are pretty much still on this chromosome. Here's one species where they've there's some that are moved over here. But we still roughly find these genes in the same spot, but the order can mix up a bit. Okay. Now, what scientists can do is they can say, okay, in a given species, compared to another species, what's the biggest number of genes that we can find still together in a block? Or, so in this case, we're comparing these two species relative to that standard species, Drosophila melanogaster. So these are two species proposed to be quite closely related, and we can say, okay, we can find, here's one block of synteny that has 1,500 genes in it that are still all in the same order and haven't been mixed up. We find, maybe about 9 or 10 that have 300, and so it becomes a distribution of how big these groups are of genes in a block. We can then say, okay, that's for these species that are thought to be quite closely related. How does this pan out as we look at more divergent species? And what we see when we do that is that we find that the average group size gets smaller over time. So now we no longer have a big block of 1,500 genes together anymore. The biggest block we can find is 150. And we can see that this distribution has shifted over. If we look at, if we carry this on and look at species proposed to have shared a common ancestor with this species much more distantly in the past, this will shift over and become even smaller, as you would predict. Questions on synteny and how that works? How do you feel about that?
The numbers
Jerry?
on the axis there,
Yep.
44 to...
Oh, sorry, a million years ago. So these species are proposed on other criteria to have been separated for about 44 to 55 million years. Whereas previously we were looking at species that were very closely related, proposed to share an ancestor about 5 million years ago. And I fully understand that age of the Earth issues are their own separate kettle of fish. I don't have time tonight to really delve into that, but the discussion period perhaps could take that on. But thank you. Okay, now that you know something about how synteny works, and you've seen how a group of species related to one another, nobody contests the idea that these are related to each other. You've seen how this works in this species. Now we're set to address the question, okay, we look at humans, we look at chimpanzees. Do they look like recently diverged Drosophilids? Or do they look like widely diverged Drosophilids? Or are they just completely different from one another? And the answer is that they look like very recently diverged organisms. So here's a high-level view of a subset of human chromosomes matched up with their counterparts from chimpanzee. So on the left, we have a human chromosome, and on the right, we have the chimpanzee equivalent. Now, we're not looking at sequence per se. What you're looking at is a DNA staining technique. So basically, it's applying a dye to chromosomes and looking at them under a microscope.